Luc Montagnier, the Nobel Prize-winning virologist known for his co-discovery of the HIV virus, is said to have ventured into controversial territory later in his career with experiments related to water memory. His work in this area, particularly his collaboration with Italian physicist Emilio Del Giudice and others, has been met with significant skepticism and criticism from the scientific community. Could you provide a detailed description of the experiment conducted with physicist Emilio Del Giudice?
The idea that recorded electromagnetic EM signals could reconstruct DNA in pure water, as claimed by Luc Montagnier and Emilio Del Giudice, is highly controversial and lacks scientific support. However, if we set aside the possibility of intentional fraud or a scam, there are several alternative explanations that could hypothetically account for the observed results, assuming they were real and not due to experimental error or misinterpretation. These explanations would still need to be rigorously tested and validated. 1. Experimental artifacts or contamination. Contamination. The water or equipment used in the receiving lab might have been contaminated with trace amounts of DNA or other biological material. Even minute amounts of DNA can be amplified by PCR, leading to the false impression that the DNA was reconstructed from the EM signals. Cross-contamination. If the same equipment or workspace was used for handling DNA samples, cross-contamination could occur, leading to the detection of DNA that was not actually reconstructed from the EM signals. 2. Misinterpretation of electromagnetic signals. Noise or interference. The detected EM signals might have been environmental noise or interference from electronic devices, rather than a true memory of the DNA. If the receiving lab was not properly shielded, external EM sources could have influenced the results. Signal artifacts. The equipment used to record and play back the EM signals might have introduced artifacts or distortions that were misinterpreted as meaningful signals. 3. Placebo or expectation effects. Observer bias. If the researchers expected to see a specific result, they might have unconsciously influenced the interpretation of the data or the setup of the experiment to confirm their hypothesis. Confirmation bias. The researchers might have selectively reported positive results while ignoring negative or inconclusive data. 4. Unexplored physical or chemical phenomena. Unknown properties of water. While highly speculative, it is possible that water has unknown physical or chemical properties that could interact with EM fields in unexpected ways. However, this would require a radical departure from current scientific understanding and would need to be rigorously demonstrated. Quantum effects. Some have speculated that quantum coherence or other quantum phenomena in water could play a role. However, there is no experimental evidence to support such claims, and they remain purely hypothetical. 5. Misapplication of PCR. PCR artifacts. PCR is an extremely sensitive technique that can amplify even trace amounts of DNA. If the water or reagents used in the receiving lab contained even a tiny amount of DNA, PCR could produce a detectable product, leading to the false conclusion that the DNA was reconstructed from the EM signals. Primer dimer formation. Nonspecific amplification or primer dimer formation during PCR could produce false positive results, especially if the experiment was not carefully controlled. 6. Miscommunication or misunderstanding. Miscommunication of results. The results might have been misinterpreted or miscommunicated, leading to exaggerated or incorrect conclusions. For example, the detection of EM signals might have been conflated with the actual reconstruction of DNA. Lack of rigorous controls. If the experiment lacked proper controls, e.g., blind testing, replication by independent labs, the results could be misleading. 7. Unexplored biological mechanisms. Bacterial or viral contamination. If the water or equipment contained bacteria or viruses, their DNA could be amplified by PCR, leading to the false impression that the DNA was reconstructed from the EM signals. Endogenous DNA. Human or environmental DNA, e.g., from skin cells or airborne particles, could contaminate the experiment and be mistakenly identified as the reconstructed DNA. 8. Instrumentation issues. Faulty equipment. The devices used to record and play back the EM signals might have been malfunctioning or improperly calibrated, leading to spurious results. Sensitivity limits. The equipment might have been too sensitive, picking up irrelevant signals or noise that were misinterpreted as meaningful data. Conclusion. While these alternative explanations provide hypothetical scenarios that could account for the observed results, none of them support the idea that water has a memory or that DNA can be reconstructed from EM signals. The most plausible explanations involve experimental artifacts, contamination, or misinterpretation of data. To conclusively determine the validity of Montagnier's claims, the experiments would need to be repeated under rigorously controlled conditions by independent researchers. Until then, the claims remain unsupported by credible scientific evidence.